good morning, everybody from Europe, and good afternoon for the rest of the path from the Asia side, I guess. So today I'm going to talk about a new platform that we have been putting together, which officially launches next month somewhere, which is RETROFIT, which is all about trying to eradicate refractory tumors via macrophage reprogramming, where we're trying to create multi-specifics that have never been created before through an AI-driven lab in the loop, a reverse translational process. I'm going to summarize a bit the science behind this platform and give a sneak peek into some of the first deliverables that the platform has created from that perspective. So, as the title already suggested, our focus in in our work here is, of course, to overcome refractory tumors. We know more than 70% of all cancer patients are essentially refractory to immunotherapy in multi-lens setting. And even if they initially do respond, 40 to 50% of the patients eventually will develop resistance, which is more sort of latent refractoriness to subsequent immunotherapy. And this is the reason why this is a very urgent problem that needs solutions as fast as possible, because there is not only the fact that there is all this refractoriness, but also that there is a big rise in early incidence of cancers, for example, like colorectal cancer. And there is also a massive amount of aging population, not just in Europe, actually, at this point, but in many other places as well. And so there are urgent need for an advancement in personalized and combination therapies uh, against these kind of refractory tumors. Uh, we have done a lot work in the last 10 years as a field, of course, in targeting tumors that have already immunogenic characteristics, for example, immunosupportive perivascular niches or pro-immunogenic or antigenic spatial hubs. Further reinforced by more immunogenic spatial zones like tertiary lymphoid structures, with the primary target mostly being the exhausted T cell trajectories that show a spatial escalation of dysfunctional gradients. And many of these processes have been the cornerstone of why the current cancer immunotherapy actually works, because you are targeting essentially cells which have been pre-exposed to the cancer antigens, and have immunoregulatory cytokines at their heart, for instance, which can be turned into something that can help the immunity against tumor if the right kind of immunotherapy is used. But in situations of refractory cancers, some of these permissive environments or factors that are good for immunotherapy response are not readily available. They are instead replaced by much more problematic situations for immunotherapy, starting with wound healing or fibroblastic barriers, for instance, that I will come back to later in my presentation, but also the possibility of active exclusion of T cells from the microenvironment, as well as actively killing the T cells in the microenvironment, all of which causes these tumors to basically not respond to any of the immunotherapies that we can currently throw at them. Uh, in that sense, there have been, of course, waves of different kinds of therapies over the years that have been used not only to tackle oncology, but also many other kind of diseases, and starting with sort of more defined molecules, but not necessarily defined targets. So broad spectrum drugs, which gave way to more targeted therapy, ultimately leading towards better antibodies in a monospecific setting. And it was widely and is still true recognized that multispecifics might actually be the future of this on a very broad scale, already from a therapeutic class perspective. And in terms of oncology, multispecific antibodies, in our opinion, represent the perfect opportunity against refractory tumors, because they overcome two of the biggest challenges that are problematic when we try to interface some of these previous drug classes against refractory tumors, which is redundancy of resistance pathways. So it's not enough to inactivate a single resistance pathway. You have to often inactivate multiple resistance pathways one after the other. So monospecifics means that you have to then chase very complicated combination therapies, which might not always be easy to chase in that particular way, toxicity concerns, but also practical clinical concerns. But this can be actually cross-connected into a single platform by having multi-specific antibodies. So this is really something that tailors the solution to the problem in refractory cancers. And last but not the least, which has been slightly unexpected but has been working really well, is the possibility to go after even undruggable targets. So, for instance, if you are talking about multispecifics that are acting more as engagers between macrophages and T cells and cancer cells, and so on and so forth, then you essentially are in that situation where you don't have to worry about whether there is a specific target you are going after. You can essentially go after just the interaction between them with these antibodies. So this has really possibilities to overcome several, if not all, refractory tumors, depending on what we are targeting. And this is already starting to show some early promise, as some of you might know, which is with this new class of bispecifics that are currently being tested quite intensively, where PD-1 or VEGF or PD-L1 or VEGF have been put together in a bispecific conformation. And we know that some of the results coming out from the work of Akizo Therapeutics and Summit together has shown that while it is early days still to confirm this, it has shown very early signs that it could be that it can compete against the current market dominant player, which is pembrolizumab. And while this still, of course, needs further validation and confirmation, these are very interesting signs for the future. But there are, of course, some problems that still need to be tackled in order for the field to actually move forward in a resolute manner. And one of these issues that are immediately apparent, if you try to kind of put the entire landscape of multispecifics that are currently being used, you see that they are very heavily directed towards, and not surprisingly, the interaction between the cancer cells and the lymphocytes of different kinds, especially T cells, but not only. And while this makes sense from many different perspectives, this actually risks doing something that has also been the problem in past, as in this still may direct most of the attention towards immune-rich tumor microenvironments that already have availability of lymphocytes, for instance, or a permissive environment to allow some of these kind of drugs to actually work. And this again isolates the refractory tumor contexts, which tend to have mostly an immune excluded tumor microenvironment or even an immune desert tumor microenvironment, which is essentially the bulk of the patients. So in that sense, many or most of the current multispecifics are actually missing, depending on the indication, 40 to 90% of the patients, which may not have very immune permissive environment or not be able to provide all the ingredients for this kind of working. And here you can always see that this is starting to change a little bit, of course, now, but still macrophages are not as heavily targeted, or the myeloid compartment is not as heavily targeted right now as the T cell cancer cell compartment. So this already needs a lot of work from that perspective. But also from the perspective of how we are interfacing our results as we go forward, from what we know about immunological pathways to how we are going to target this in the clinic, we have more and more appreciation of the barriers that are there, and these still need to be properly interfaced with the field of multispecifics. So, for example, a lot of how the multispecifics are currently handled is about to start with a lot of the omics technologies, paving way for what are the kind of pathways or target combinations that we have to take into consideration, what is the possibility of their success. But some of these steps are still ignoring some more critical bottlenecks or barriers that exist in the field. For instance, organ-specific immune networks is one of the most important barriers, because while eventually it is possible that a new therapy can be positioned in earlier lines of therapies for certain patients in late clinical trial success based, But in the early clinical trials, phase 1, for instance, we will mostly be talking about patients that are heavily metastatic and or have been heavily pre-treated previously under a metastatic disease. So this essentially means that we have to confront the organ-specific barriers upfront if the drugs have to pass a more critical phase 1 barrier with this more mixed or basket trial situation, sometimes even. And this is sometimes not taken into consideration. A lot of our work early on tends to be about tumors that are going to be permissive to multi-line therapies or early line therapies. And not to forget, very close to that are, of course, evolutionary barriers and divergences or convergences in the immune system. While the road is leading towards human application, the most of the work, of course, early on will still remain a lot in model organisms. Even if we have alternative models for human representation, which is an important trend, it would still be a combination of different approaches, which means some of these evolutionary barriers, they also need to be already taken into consideration when we are creating multi-specifics, more so than even monospecifics, because if a certain multi-specific combination ends up being model dependent, then this will have harder problems when it comes to translatability. And last but not the least, something that has plagued almost all immunotherapies after a certain point of time is accountability for population immunogenetics. This remains difficult for most modalities, so it's multi-specifics and not an exception. But considering that for most part of the cancer immunotherapy revolution, uh, the understanding of omics technologies and the data has kind of accompanied our understanding of how these immunotherapies are currently working in the clinic. It is quite opportune to already, for the next so-called revolutionary therapeutic solutions like multispecifics, that we start taking into consideration the population immunogenetics upfront. And last but not the least, here also we have to start learning lessons from what has already passed. And in that sense, we have to also confront that when it comes to modeling or preclinical testing, that sometimes in past, the threshold for what we consider as preclinically valid has been somewhat relatively low, depending on who were working against it. And while here I will use TIGIT as an example of this. Some of these shortcomings are not necessarily limited to TIGIT. Many other targets like TIM3, VISTA, CS7R, TREM2 have had similar fates in their handling. So, for instance, when I say it has been lower threshold for preclinical validation, one of the example here would be, for instance, that if you're looking at murine validation of some of these antibodies, a lot of times they are only working in a combo with PDL1 or PD1, but by themselves as a monospecific efficacy, there was little or no efficacy demonstrated for many of these targets in the last decade. And so this is already a big bottleneck, which meant that by their default, these antibodies did not capture some of the pathways that they were supposed to capture, but only in combination, which really meant that now additional barriers were applicable. Moreover, some of the mouse models that were used and are still used quite heavily for validation were not necessarily selected based on the human tumor landscape, which has to eventually connect to these therapies. And it was also not always clear whether the mouse models were recapitulating the human target or MOA biology. So, for instance, for TIGIT, while early on it was always considered to be a compensatory checkpoint, second or third in line to PD1, uh, suddenly later preclinical evidences started moving towards a role for myeloid cells, or macrophages, while this was not necessarily taken into account in the early steps of development of this drug. So these, if you put all of these together, it does paint a picture where definitely there are a lot of problems with target selection and target engineering. But when we start criticizing the preclinical validation of immunotherapies, then also it has to be critically analyzed whether it was always done as well as it should have been. So one of the solutions that we think in our lab and this platform that we are starting to come up with is that apart from all the revolution of trying to have human equivalent models, we should also seriously consider reverse translation based methodologies to be present in the early development of therapies before forward translation is taken into consideration. So what this very simply means is that all the decision making in preclinical setting or experimental setting, uh, testing in mouse, is fully defined and guided by human data rather than the other way around, which happens a lot. Start with experimental systems of mouse, and then you try to validate something in human, and then we hope that the forward translation follows the same route. Instead, the reverse translation suggests that you start the problem as completely human based, and then try to see how much of it and what of it can you translate to different models. This obviously means that you are confronting many different dimensions altogether. Initially, this does put you primarily in the domain of conserved pathways between species, where are essentially target biology, mechanism biology, omics data, as well as immunophenotypes have all come together in order to help prioritize which kind of pathways you are going to go after for reverse translation. The pathway dimension can be increased, of course, by maybe testing with other animals if required, which is also happening now little by little. But as we know, there is a whole revolution around non-animal ex vivo or in silico approaches, which can also accompany in order to increase the dimension of what we are testing here. So it could be that you can replace, for instance, mouse with some of these approaches. But again, depending on what kind of therapy you are dealing with, this may not be always so straightforward. So, for instance, if you're dealing with something that is so systemic as immunology based drugs or immunotherapy based drugs, then you need the host immune system to be recapitulated. And this is not so easy with organoids or even explants, for instance. All the work is going on in that direction. And so if the therapies are more directed towards cancer cells, or then organoids, for instance, can be quite well used, or PDX models can be quite well used. So these choices here will help expand the pathway horizons, but it's already for conserved pathways that we still need a lot of work. As we know, after PD1 and CTLA4, others have not quite worked out, even though many of these pathways exist both in humans and mouse. So I believe that even for the conserved pathways, we have not properly done all the work yet. And the idea is that all of this is, if this properly accomplished, can essentially start going towards forward translation in terms of innovative immunotherapy or biomarker solutions. Now, this looks good, of course, but there are still hurdles for this kind of approaches to go forward because, especially when you're talking about translation towards the clinic, which also then eventually involves translation from an industrial perspective, then it has to consider a lot of constraints that have to do with R&D productivity. So you need to achieve also reverse translation at a sufficient R&D scale in order to balance effectiveness and efficacy. Now, this is exactly where our work has been located in the last few years, and this is the platform, so-called RETROFIT, that I'm talking about, that we hope to launch in the coming months. And the idea here is to actually take a road towards first or best-in-class multi-specific antibodies by going through and integrating all these different layers that try to confront the different barriers that I just talked about, all the problems that are there in the state of the art, starting with multi-omics and systems medicine based approaches that are feeding off of cross-species data lakes that are then cross-connected to AI-guided target discovery. This will then help you create rational antibody libraries because you can then have the right combination, the right target, because you have pre-informed this through a human-driven as well as a cross-species-driven approach. And this approach also then allows you to rationally select the disease model. So you're not randomly selecting your mouse models, but your data is telling you which human recapitulating models should essentially be connected to the target that also came from the same data. So this creates a very nice synergy between the development of a target and a therapy, as well as the models you will eventually test them in. This, of course, also creates challenges which is essential in my opinion, because sometimes the model, even after doing all of these processes very well, the model may not actually work out with a certain immunotherapy, which is a very good indication that you have to further work on that pathway. So there is resistance from that model, which was otherwise very neatly connected to that target. And if it didn't work out, then there is a huge reason here to push forward, especially if you want to keep developing the pathway, because then you have to assume that the model is recapitulating also human resistance biology properly. And I will show you that in a bit, how we are able to accomplish that in our own setting. Finally, this also means that you're cross-connecting this on a functional immunology level. Here, you can already start going, for instance, for cross-species validations. For instance, you don't need to be restricted on functional immunology level to mouse models anymore. You can already start interfacing human alternative models with mouse models, because essentially you have MOA from mouse already, which you can start translating into human systems. So this is why you're translating some of the mouse observations already into human setting. This can then connect to some higher level of translational parameters. For instance, the preclinical human models that are more advanced, like explants or organoids, where you can do more final sort of validation, which is already starting to gain quite some traction with approval agencies in FDA and beyond. And finally, lastly, you can cross-connect this to CDX methodologies like spatial or digital pathology biomarkers. All of these layers is what we have been working on in order to lead from a systems medicine starting approach to ultimately a first or best-in-class multi-specific antibody. But and there is a lot of competitiveness that we have baked into these different steps. So, for instance, there is a lot of privileged data access, unique multifactorial data analysis methodologies, proprietary annotations, and unique target discovery approaches, which are fueling the competitiveness of some of these steps. Ultimately, we also have several non-obvious composition of matter patents possible from this library, which is an important select factor going forward from a translational perspective. And the good thing is that even though this is oncology focused, eventually we can also use the same data to diversify towards other diseases. This is something we are already doing, because now you know the pathway, you know the context, so you can use the biology to go to other diseases in a more rational fashion. So has this whole system come together in overnight? Not really. We have been working over many years on this, and the optimization, priming, and stress testing of our system actually followed an approach that is relatively similar but not identical to what is proposed to be a lab in the loop or a clinic in the loop model, where you essentially talk about starting with a certain data which may or may not be completely part of a foundational model. So for us, for instance, it's not really one all-encompassing global foundational model. It's many different gating components and a lot more, for the lack of a better term, Boolean gating like strategies rather than one foundational model. But of course, the basic idea is that in certain cases, you can use the data to feed a single foundational model, which then supervises some of the experimental choices that you are talking about, essentially a way of doing reverse translation, and then feeding this back towards the data. And this can keep spiraling until you can optimize your pipeline towards a correct target or drug. So this was an inspiration for how we optimized retrofit as well, but was not necessarily identical to it. So I will, in the next slide, walk you through how we actually did it over the years. So we started off initially with optimization of multi-omics and spatial omics-driven systems immunology. So here we had multiple publications, which are also in public domain, that allowed us to optimize some of the early data lakes and the omics analysis methodologies. This allowed us to undertake our first reverse translational pathway prioritization approach. So here the idea was, how can we use reverse translation for prioritizing certain pathways? This loop actually ended up producing its own deliverable, a first-in-class serum screening assay that we have been using and has already been clinically validated for multiple patients for ovarian cancer, lung cancer, head and neck cancer, and breast cancer. in both retrospective as well as prospective setting. And there are other clinical studies currently ongoing. So it really proved that when we undertook this loop, that it could actually convert into a clinically tangible deliverable in the form of an entirely new biomarker assay, which also was eventually clinically validated. So the already first proper reverse translational loop was quite successful, which was quite heartening. So we decided to take the next step, use the same reverse translation but with AI in order to now inform combinatorial immune checkpoint therapy designs in the clinic as they are in the clinical standard practice right now. So here again, we were able to create a new clinical deliverable for the first time, an AI-based neoantigen specific HLA biomarker, which I will describe to you in the next slide how we did it. This is currently already part of testing with patients with kidney cancer, but also even glioblastoma as a diversification strategy here. We have clinical studies ongoing mostly locally right now here in Belgium, in Leuven, based on this AI design biomarker, which is telling us a lot about how the patients should be handled. Some things that we didn't even know when we created it. So really a good example of how some of the things that AI learned that we didn't know it was initially learning is now informing some of our choices. So now that we were able to also apply this AI-based reverse translation for existing checkpoint therapies optimization in the clinic, we took the next step and went even a further step ahead, even a bigger challenge, which was to use reverse translation to now design anti-cancer vaccines. And this was also extremely successful in the end, because we were able to confront the challenges of the dendritic cell vaccines that are currently being used, optimize them using reverse translation, and actually found that it's not just enough to have a very nice dendritic cell vaccine, which is what has mostly been the focus of the research for many labs in the last decades, a very, very immunogenic and a very good vaccine, but also it has to be rationally combined with certain pathways, because it doesn't always work with every generic pathway. In this case, the surprise was not PD-1, but only PDL1. So this was very interesting, because one is very tantalizing to just go and combine a vaccine with PD-1, which is clinically applicable and broadly a good immunotherapy. But we found PD-1 was not working. It was actually really PDL1 because of a specific reverse translational pathway analysis that showed us that this was the specific pathway. And I'm also very happy currently that even this as a strategy is currently undergoing clinical validation and substantiation. And this is actually international right now because the clinical studies are ongoing in Germany and Norway, funded by the prestigious European Union's Mission Cancer grant. So this is a bit in nutshell, four major lab in the loops cycle that we took into consideration. Every single cycle over the last several years has essentially optimized one or the other component of this platform, so that and the success of this cycles is actually basically judged by the fact that every cycle was able to create a tangible clinical deliverable, which also either already got clinically validated or is under high impact clinical validations currently. So this is a bit in a way for you to appreciate that the loops are not only useful for optimization, but they are also cross verifiable as successful loops. So how does a loop exactly look like? And I'm going to use this example, which we published in Nature Medicine a couple of years ago, where the problem started essentially from kidney cancer patients. So for those who may or may not know, while kidney cancer is amongst one of those so-called hot tumors that are supposed to respond really well to immunotherapy, it shows quite insurmountable heterogeneities that do not make it a one-to-one positive correlation between how immunogenic or inflammatory it might look sometimes and how much immunotherapies actually work out. There's quite some massive heterogeneity there and very huge amount of questions that have not been properly answered because the mouse models for kidney cancer have not been as good as some other modalities. So you are a lot more reliant on human data than some of the other fields where there are more mouse models available, for instance. So one of the challenges for us starting with this disease here with the clinicians was that can we actually decipher the heterogeneity of these patients in order to understand what really defines a responder kidney cancer patient and what differentiates it from a non-responder kidney cancer patient. And based on that, can we propose some rational combinations of where do we have to go next? What kind of combinations should be defined based on the data beyond what is in the clinic right now, which is primarily PD-1, CTLA-4 combination therapies. So here we did this multi-omics pipeline that I was talking about. I'm just giving you a snapshot of it, but actually from a spatial perspective, but we did almost all the omics resolutions, bulk transcriptomics, spatial omics, single cell omics, as well as spatial proteomics. But what we found was the patients who were responding well to immunotherapy in the clinic, in the real world setting, so really outpatient real world setting and not just a controlled clinical trial, was a very specific antigenicity signature that was cross-connected to a complicated spatial community, capturing interactions between pro-inflammatory macrophages and exhausted or pre-exhausted T cells. And so this signature was really differentiating a responder phenotype. We actually then converted the signature into something that was a lot more AI friendly for machine learning analysis, and were able to validate that indeed across nearly 1,000 patients, several clinical trials, both in-house as well as published, this biomarker was indeed differentiating patients that respond better to clinical immunotherapy, PD-1 CTLA-4 or PD-L1 alone or PD-1, all three flavors. And we could differentiate objectively patients who then do not respond properly to current immunotherapy in the clinic. Then, of course, the idea was, as we said, from a reverse translational perspective, that, okay, from a human perspective here, we have a signature, we have the biology under our hand in terms of what differentiates a responder from a non-responder. Then can we do more here and actually try to see to move forward towards new targets and new models? So this is where we did reverse translation. We actually took a human signature and applied it in mouse models. So this is a human antigenicity signature in mouse models, arranging many different mouse models that are well known to us in the order of their enrichment for the signature. And interestingly enough, this signature immediately picked as the topmost hit Renka, which is the only transplantable kidney cancer model currently being used in the clinic. So this was very surprising for us and very unexpected, because while we did hope that we were open essentially to the possibility that any model, irrespective of being a kidney cancer model, could be a top hit, and then we can take it into consideration as a reductionist model rather than being orthotopic model of that disease. We were pleasantly surprised that reverse translation was quite successful, that a human kidney cancer signature immediately, in an unbiased manner, picked also a mouse signature. So this kind of shows to you how powerful reverse translation can be if it is properly done, because these kind of approaches for model selection are very rarely used. And then what we did was we went a step forward. We, as I said, we also did a lot of single cell analysis here, which I'm not showing, which allowed us to have certain targets that were supposed to be predicted as the second in line to PD-1 combination that could be useful in the patients. So what we did was we then used these different targets which were predicted, which was TIGIT-LAG3, as it often happens with these kind of settings, but also CD40 agonist, which is a myeloid target, as we all know, which is an activating target, essentially talking about CD40 agonism, which will allow activation of the macrophages. And that is essentially what we found was actually the best combination of the three that we tested. That while PD-1 alone was working, all the other combinations were not superior to PD-1. In fact, had chance that if you combine them, that it might make the efficacy of PD-1 worse, which is something which has been seen for TIGIT actually in some trials. So this did recapitulate what we has been seen in the clinic. But the best condition was the one where PD-1 was combined with CD40 agonism, again connecting to the mechanistic observations in our data that it is about macrophage T cell community, and hence you need to further enforce, for instance, macrophages while you are reinforcing T cells already with PD-1. So a combination of PD-1 CD40 agonism. This is currently, as I said, subject of further development, because this AI machine learning algorithm and biomarker that we created is now giving us further insights into what makes immunotherapy tick in kidney cancer patients and what works and what doesn't work. So this is a very nice example of a lab in the loop we have already done. And as you can imagine, all of what we have learned here is what exactly we fuel into RETROFIT platform right now, which arranges all of this information again and again in different loops. So what are we busy with right now as we speak are essentially two very, very innovative lab in the loop cycles, which are going to further not only optimize the platform, but again, as has been the nature of all the loops we have done so far, also give us some of these new deliverables. So for instance, these are the two loops we are currently ongoing, a reverse translational approach for target discovery for multi-specific antibodies, uh, together with a spatial omics and functional immunology approach in order to create proper digital pathology modalities. So, what is our target for all of these new efforts right now? And we have a very specific target there, which is one of the most pervasive targets, which is wound healing or fibroblastic barriers in cancer. For those who have worked with refractory cancers, they would understand that this is something that is a massive problem for a lot of tumors, uh, where there's a lot of wound healing going on of fibroblastic barriers. And interestingly, targeting this niche also gives us very high disease diversification possibilities because problems with wound healing are also common in some other diseases that have nothing to do with cancer. Because this inability to heal a wound and for it to become festering wounds or something that doesn't resolve on its own is shared by many immunopathologies, for instance. So that's why we went after this sort of a more broad pathway because it has been also traditionally difficult to take care of this in oncological context. Beyond something relatively associated to VEGF pathway, there is not much other options around the wound healing paradigm. Now you have to consider why this is so, because wound healing paradigm is not really very easy. It's a very complicated process. And if you really look into it or are working on it, you will realize that it actually affects almost all or many of the so-called hallmarks of cancer. It's a bit of a silent player, or as I would like to call it, a sort of a puppeteer in the background, which actually fuels a lot of different problems in cancer. Wound healing causes induction of angiogenesis, which is uncontrolled, which is why I said that VGF is probably the only one that has been recently looked into from this perspective. And then there is a lot of activation of invasion and migration of cancer cells. And wound healing also promotes chronic inflammation, which is then cross connected to a lot of different pathological parameters in cancer. So, your fat signaling, microbiome dysbiogenesis, matrix deposition, which is uncontrolled. And all of this then fuels several of the other directly or indirectly hallmarks of cancer. So it is really a process that has been sometimes ill-defined, sometimes defined depending on the context, but actually cross connects to a lot of the work that is ongoing in different hallmark categories. Here, one of the important barriers to T cell infiltration and immune infiltration is, of course, matrix deposition. And this is where comes this sort of a collaboration between different problematic immune cells that fuels this. So this is a multicellular community problem. It cannot be solved all the time by a single modality. So you have lymphocytes or T cells, which are essentially using a lot of type 2 cytokines to cause these kind of matrix depositions. And macrophages then collaborate on that level by producing TGF beta or matrix metalloproteinases, which also cause invasion and degradation. This together actually recruits a lot of fibroblastic signaling, which puts together these fibrosis-like barriers inside the tumor. And this might sound familiar, especially for those of you who might have a medical background, uh, that fibrosis is not just a problem in a tumor context, but fibrosis is also a major problem in other pathologies, as I just said, for wound healing. Uh, for example, diabetic foot ulcer, some of the autoimmune problems as well might be cross-associated with fibrosis. And there are also some obvious wound healing issues per se by itself, where where the patients have likelihood of creating fibrotic wounds that keep expanding. So again, as I said, it has an oncological impact, but it has also a non-oncological impact in long run. But the problem is that this is a more simplistic view of this, especially if you think about from a macrophage perspective, which are really strong drivers of fibrosis due to the TGF beta production. Because as is also apparent for a journal like Myeloid Cells here, macrophages is not a single entity, as we have understood. And while for a long time, due to mouse only research, it was simplified under the umbrella of M1 macrophages, which is pro-inflammatory, or M2 macrophages, which is anti-inflammatory. This, due to the advent of advanced single cell technologies and spatial technologies, has now been strongly challenged, as we know, and rightly so. But that basically means you have a huge anarchy in a way, because now, depending on which context, space, or environment you're looking at, you have many different kinds of players which might be contributing to these processes. Many of those might be then cross connecting to causing or this kind of fibrosis, or even dysregulating this kind of fibrosis. So it's very important now, going forward, with all this information that we know about the complexity of the macrophage compartment in the tumor, to really understand how are we actually going to go after this. What is our best approach to therapeutically eradicate multifactorial wound healing landscapes? And this is the right kind of problem we felt for RETROFIT, our platform, because this layering of information and this complex workflows that we are creating are the best way to be stress tested or challenged with this problem, which is indeed what is the biggest problem for refractory tumors right now. So we thought this was a worthy challenge for us. And that is what our platform has been doing. We essentially have three candidates in development right now. The top candidate we have is actually a highly novel tri-specific antibody that cross-connects VISTA, TIM3, and CSF1R. So this is what I'm going to briefly talk about today, which is in the most advanced stages of preclinical development right now for us. But we also have, in the same perspective for wound healing, many other kind of therapies that we are also working on, also from a sort of a T cell fitness perspective, but also other TME resetting perspectives, which also cover multiple malignancies. So TREMA is patent protected right now and is under valorization from this platform. So that's what I will briefly talk about. But before I talk about how TREMA actually looks, it might be interesting, again, from a lab in the loop perspective, to see how did we actually come to it. And again, we did not target the macrophage of interest immediately, as is the tendency to do for many people, because, as I said, it's a complicated environment, macrophage in a tumor. So it's important going forward, if you're developing a therapy against it, that you know that you're really targeting something that is relevant to the biology of the tumors that you are dealing with. So what we did was, in order to understand this, we took single cell signatures of 26 different tumor associated macrophage subsets, and confronted them with a huge amount of data from TCGA, and cross-connected it to the different spatial or molecular landscapes that have been validated in TCGA dataset as well. So this is interferon gamma dominant, of course, and inflammatory, which tend to respond to immunotherapy very well, going towards lymphocyte depleted TGF beta response, immunologically silent, and wound healing, which are more complicated and heterogeneous when it comes to immunotherapy response. And then when we cross-connected the different macrophages to this situation, we actually found that the macrophage that was most strongly associated with wound healing, which was the biggest target for our platform, was actually the TIM3 VISTA positive, double positive macrophages. So this really shows to you a sort of an unbiased way of approaching this from human data perspective. So while there's a lot of macrophage targets possible here, and many of them have merits, from a biological perspective, for example, many of the macrophages you might see here are very well known and worked upon. At least from a data perspective, it was clear that if we have to go after wound healing, we should probably prioritize starting with these macrophages. But we didn't stop there. And this particular pipeline will show to you how RETROFIT actually works as an approach, in the sense that we start then again with human data. But TCGA, as you know, it doesn't have immunotherapy responder, non-responder information. So we actually took a lot of different clinical data from patients that do actually either respond or do not respond to immunotherapy, and reconfronted it, which allowed us to actually get to some of these macrophage subsets which have a bad impact on PFS under immunotherapy. And of course, of all the different landscapes, it was indeed wound healing and not some of the other landscapes here, which was the problem. Wound healing was definitively a negative predictive factor for immunotherapy in patients. And together with it, there were many other macrophage subsets that were also important. Then we did what we keep calling reverse translation. We went to mouse, but we did a specific step which a lot of studies do not do, uh, in my experience, 95 to 99%, which is to also do the responder non-responder benchmarking in mouse. So most of the mouse reverse translation, even if it is done on data level, is done from the perspective of treatment naive tumor models. So you go from all the human data to essentially no idea of whether you are dealing with a tumor in mouse, which is responder non-responder, but just a model resolution, which may not give you a full idea. But here, what we did was then we actually took responder non-responder mouse into consideration, and we also took into consideration the species-specific immunology. So while here the immunology of macrophages was obviously a lot more about subsets and their alignment with special landscapes in human tumor, which is exactly the biology of macrophages in humans. In mouse, we know, of course, that this biology is a little bit more simpler from the perspective of M2 or anti-inflammatory macrophages versus M1 or pro-inflammatory macrophages. So again, when we pass the species barrier, while we did some benchmarking or overlapping on the level of responder non-responder, we also accounted for the species-specific biology, so we can tailor it to the landscape situation. And actually, when we did that, we found that one of the top hits was the TIM3 Vista double positive macrophages, uh, which had the highest association with anti-inflammatory environments in the non-responders as compared to responder mouse models. And interestingly, when we looked further, we actually found that there were a lot of different models that were associated, that were fueling this data. And many of these models are not actually used that frequently in the oncology setting. So this also tells you something. We found LLC model as one of the more benchmarkable model from this list. Many people use it as well. That's why we selected it in order to allow better comparison of our results. And further single cell analysis actually showed that indeed we can find specifically this TIM3 Vista double positivity only in the macrophage compartment. And further work actually showed, which I didn't label here, is that this was under the CSF1R lineage. So here it is very important to consider that many models that are used mouse-wise in the field for macrophage biology analysis, even sometimes like MC38, B16, CD26, 41, all of their data from this perspective of responder non-responder was inside our atlas here. But when we did this rationally, these models were not driving these results for immune refractory macrophages. It was these models instead. So this again comes back to the point I was trying to make on the slide of the digit, that have we always done preclinical research with the models that actually recapitulate human biology? Because if you do try to do it rationally, you do see that there are exceptions to their use. And at least in this particular instance, uh, different models were actually recapitulating the original biology rather than the one that you might sometimes think is cold and could be used from this kind of a list. So this is one example. In this particular case, this is applicable. It might not be in all the cases, but at the very least, it shows to you why it is important to do reverse translation in a data-driven manner. So we also did then further validation of this work. We found the macrophages, of course. So we also did a TIM3 Vista CRISPR-Cas based knockout in the CSF1R lineage of these macrophages. And we did see in this particular model that the knockout of TIM3 Vista by itself was already having an impact, which was even further improved if we combine it with immunogenic chemotherapy, in this case, paclitaxel. But the impact was still relatively modest or limited, at least from a CRISPR-Cas9 perspective. So then we considered that maybe is the way we are targeting these macrophages has to be changed from this perspective. And now I'm not showing you the results we did with monospecific immunotherapies, but it was not as easy there either. You cannot just target them with a TIM3 antibody or a Vista antibody and it would work out. In monospecifics, they were not working at all. This is where actually further ideas came for trying to create multi-specifics, and I will not go into the detail, but essentially we had a lot of data that suggested that these pathways might be following something that might be well adapted to targeting via multi-specific antibody rather than trying to do it with a monospecific. So that's what we basically started off with, and created a tri-specific antibody against TIM3 Vista and CSF1R. And we tested it in this LLC model, which is essentially a preclinical representative of a tumor mutational burden low lung cancer model. But we didn't go immediately just to test our antibody and show that it works really well. We also redid a benchmarking of the model to align to what we have been saying so far. So I have been telling you so far that we want to confront refractory tumors, tumors that do not respond to any of the immunotherapies that are already there. And so it was necessary that we also benchmark this, which is also often not done. That is your model recapitulating the problem you're trying to solve in the first place. So we did that, and we first did for the first wave of checkpoint therapies that have been used against T cells. And indeed, this model was resistant to everything, not just the PD-1 immunotherapy, but also the combination of it with many different players, which have been significant players of combinatorial landscape in oncology in the last 10 years. And indeed, this model did not respond to it, which means it was refractory indeed to all the immunotherapies that have been popular lately. We also pushed it further towards a sort of a so-called second wave of IO monospecific antibodies, which went after more stromal pathways or macrophage pathways, including CD40, for instance, which were all supposed to treat them already in monotherapy settings in principle, because these kind of tumors do have many of these different pathways in it. But again, the monospecific setting, there was no difference in this tumor working in any particular way. Again, proving these tumors are indeed, as the data suggested, extremely refractory to immune targeting. Then we went a step further and actually targeted the third wave of IO therapies, which are currently in solid tumor oncology, the bispecific antibodies targeting the PD-1, PD-L1 VEGF pathway, which is, as I said, comes a little bit closer to doing something with wound healing, because the angiogenesis is an important part of wound healing dysregulation. And this has also been one of the selling factor for the bispecifics VEGF, PD, and PDL1. And indeed, we see, as it has been suggested, that these bispecifics might be actually promising for these kind of refractory tumors. And we did see a probability of around at least 40% tumor eradication as monotherapy for PDL1, PD1, VEGF bispecifics, which is indeed a sign that at least with these bispecifics, the field is starting to move towards a good solution against this tumor. But you would agree that this was still not really impressive enough. This is when we actually went to TRIMA, which is an obligate trispecific. By the way, you really need the three targets to be targeted with this antibody. And it gave quite some surprising and highly significant results. So while if you just give the monospecifics, as I said earlier, for TIM3 Vista or CSF1R, it doesn't work. If you just combine them like a triple antibody combo, it also doesn't really work. But by putting the three specific antibody together, we already achieved more than 70% eradication as monotherapy, which was higher than any other monotherapy efficacy you can see here, which not surprisingly got even better, more than 90% tumor eradication as combination with the standard of care anti-PD-1. So this kind of really shows to you the power of reverse translation through the RETROFIT platform. You really select the targets tailored to the right biology and then test them also in models that are recapitulating that biology. So essentially, what we have seen so far is that these macrophages are quite enriched in tumor mutational burden, low lung cancer, and also MSS colorectal cancer. And if we give TRIMA, we essentially start resolving the wound, which causes something that has also not been reported before for any immunotherapy, at least myeloid immunotherapy in the last 10 years, which is it does not only do what one would expect is to activate the cytotoxic CD8 plus T cells via type 1 interferon signaling, but it also uses an antiviral mimicry like activation of TAMs to activate cytotoxic TAMs. So now you're not only waiting for recruitment of cytotoxic T cells into the tumor, but in the meantime, you're already starting to reprogram and reduce the burden of the neoplasm by cytotoxic TAMs. I think this is an important reason why this trispecific antibody works so well, because this very unique dual anti-cancer cytotoxicity mechanism ensures that while the latency of this process is taking place, because we know after a new activation of antigen-specific immunity, it takes some time for the T cells to come back into the tumor and start reinforcing some of the processes for anti-tumor immunity. But in the meantime, an aggressive tumor like these ones might have already moved away. They may have already reduced the antigens that the T cells were originally primed for, or they may already start putting up barriers against their against their infiltration. Which means even if you do this process properly, you activate the cytotoxic T cells properly, by the time they arrive back from the lymph nodes, they may not able to enter these tumors because of the new fibroblastic barriers they may have created in the meantime, uh, or that they have already started to change their antigenicity, which happens a lot, which is what is then taken care of by the cytotoxic TAMs. They keep the local reaction going, avoiding the creation of these barriers by resolving the wound and by keeping the antigenicity high through the cytotoxic effects, which means that then they eventually create a position of very nice collaboration between them to overtake these kind of tumors, which is essentially what you saw in that slide there. Extremely challenging tumor that doesn't respond to anything, that just targets this axis or this axis, but then really brings these two things together to create a dual anti-cancer cytotoxic effect. This is extremely novel, and in my experience at least has not been demonstrated for any immunotherapy in the last 10 years at the very least. And we also see, of course, that the signatures associated with macrophages are actually correlating with immunotherapy resistance in patients. So we have association of human biology with this mechanism of action. So I hope you can appreciate that this actually has our approach with retrofit overcomes a lot of barriers that are sitting at the valley of death between preclinical research and industrial or clinical translation. And if we do it properly with retrofit-like approach, you can not only achieve combo efficacy with standard of care immunotherapy like PD-1, PD-L1, but you can also have monotherapy efficacy, which has been one of the biggest lacking factors for many of the data packages for immunotherapy in the last decade. Uh, and then you can have a mouse model that is actually really selected by the tumor landscape of the humans, which essentially bridges the gaps between them. I'm not saying it completely overcomes them. That is impossible because there is 75 million years of evolution that differentiates us. But it does actually allow a better bridging of the pathways. And finally, we are also working on now mouse models to see if they are recapitulating human target or MOA biology. So this is ongoing, where we're making the reverse bridge. You're using the human models now in order to connect them to mouse models and see if you are able to get the same MOA biology in both species. This is ongoing, and the early signs are that this really works. Last but not the least, as I earlier said, that there's a second loop we are working on in retrofit, which has to do with digital pathology in order to be able to select patients. Because when you go into the clinical trials, you need to be able to select the patients so that we can actually go forward in terms of clinical translation when the therapy is ready, and we can do patient selection and stratification in long run. So this is what we have been working on at this point of time. And this is our pipeline for this purposes that takes into consideration all the markers of macrophages that we have created for this particular macrophage subset, that we are able to basically use the HNE, which is routinely done in the clinic. So that's the principle behind digital pathology. You use image-based initial analysis of these macrophages. You do patch extractions from existing HNE stains of the slides that are found in the clinic, so that you can then do deep learning kind of analysis to predict where these macrophages might be in tissue samples that are HNE-wise clinically highly accessible. So this is very efficient and cheap to execute in the current practice without requiring heavy IVDR and biomarker approval work that otherwise is a problem. For example, this is how it looks like. You have two images here, which are standard HNE images of the patients, which have been digitally stained for the macrophages, so they can really show where our macrophages are located in the different tissue. Uh, so this is really digital staining, not actual staining. So this is really fantastic from that perspective. And it does also capture the biology that we are dealing with. So for instance, if you extract these patches and look at what kind of pathways are actually working here, you can see that you have a lot of wound healing pathways like IL-6, STAT3 signaling, which is enriched in these patches where this is. And this also allows you to essentially have prognostic impact already, which means these patches are working. Because when these patches are low, the patients are responding better than when these patches are high, which also aligns with some of the biology of these macrophages around immunotherapy resistance. So really now you are able to translate the biology you see on signature level and mechanism level onto the H&E, and it really works as it should in terms of mechanism of action. So this is really fantastic, and this is again where reverse translation is very important, because the signature is connected with certain MOA, and then you can cross-check that MOA for different pathways and prognostic analysis to really connect now your biomarker and patient strategy of selection also to the biology. So this is also very unprecedented and often very hard in the previous classical ways, because you cannot always recapitulate this with simple plex immunohistochemistry, because it may not capture all the pathways that a signature can capture for that entity by those three proteins. And the additional addition of antibodies also creates problems, as some of you might know, with PD-L1 antibody. So currently the platform is actually busy valorizing this antibody further towards an industrial trajectory. So we're also doing all the human validations, as I said earlier, initial IP filings are done, and we will also do more advanced IP filings eventually, so that we can start creating a clinically nominated candidate by the end of next year, which is all very much on speed. So we are very happy about these developments. So where are we going next with this particular platform right now? We are trying to confront also all the headwinds that are currently there in the field. So there are other 3 lab in the loops that our platform is currently doing. For instance, we are trying to now convert many of the things we have been doing into a multi-dimensional agentic AI for target discovery and recommendation. So some of the things that we have done now on a case-by-case basis, or rather more human level, can actually be automatized with an agent AI, which can do things, at least some of the things, more automatically, if not everything. And then we basically have a very important headwind in the field right now, which is AI-driven de novo antibody design that we are going to be working on as well, to interface with everything we are doing, so that we can find hits and select them in order to have the antibody designed entirely by AI for the targets we find. And finally, of course, also to interface human biology better with modeling. This is something which is a challenge for everybody, and we will also confront that in our setting. Essentially, the idea is that when we progress further in the next coming years, that we can create an end-to-end AI-driven target to antibody designing strategy, which can go really from a target to situating this in a clinical setting in a very looped fashion. And the ambition is to address this valley of death in terms of therapeutic development between fundamental research and industrialization, going from our setting, which is the university, of course, and interfacing with the clinic and the industry. That this is one of the biggest problems, and hopefully, if this loop is done properly, that we can reduce this sort of bottleneck for therapies, and increase their developmental efficacy and efficiency. At least we hope that. And the idea is to initially confront this by putting our therapies into a spin-off, which we are also working towards. And this will be one of the first examples for us to address this valley of death with this startup that hopefully can be created to valorize, for instance, Trima that I just talked to you about. This is the RETROFIT team, which is currently leading this. We have multiple experts who are part of the founding committee. I am the director for this, and we have also a very expanded and competent management team, a business manager arriving very soon. So we are very excited about this, and of course, I would like to acknowledge as well all our lab members, collaborators, and funders for all the help and work over the years. So thanks a lot for listening to me, and I'm happy to take questions.